this is going to be sort of a review of everything we've done in the unit. Starting with just the basic Punnett squares. We're just going to make a list of everything that you should know, and I'll give you an example of all the Punnett squares. And this, I think this will be helpful to really just go back and make sure we understand all the different Punnett squares we talked about. And those of you that have been absent, I think this will at least show you, again, what you've missed and what you may need to come in and get help after school for. So just write what I have on the board. The first type of Punnett square is called a monohybrid cross. A monohybrid cross. There's 6 Punnett squares that we've done. 6 of them. That's why I think it's important to come back and just briefly, spend 10 or 15 minutes making sure we understand the difference between all 6 of them. So a monohybrid cross is a single trait. We're talking about a single trait. That's what the word mono means. So for instance, this would be like saying we have tall and short people. All right. One trait, height. Plant height, human height, dog height, tall and short. So if we have, let's say, a homozygous tall person, big T, and a short person, little t, we would want to know what their offspring might look like. So if this is the cross, and I know this is the very basic genetics that we've done. So I know that to this point, you all get this. Right? I know that. So if you did this Punnett square, of course you'd be big T on the top, little t on the side. Doesn't matter which way they go, what goes on the top and what goes on the side. No, it does not. But you would notice, and I think I heard someone already say it, is that you get individuals that are all tall. And that's true, but that's only one part of the data that you'd be responsible for. I'm pretty confident that you all can do Punnett squares up to this point. Where we all seem to get lost are the differences between the phenotypes, and this was with all the Punnett squares, and the genotypes. The phenotypes are what they look like. So someone already said it, tall and short. So someone already said that they'd all be tall. Why would they all be tall? So the big T, at this point, with these Punnett squares, if they have the capital letter, they're going to show the dominant trait. At this point, that's what that means. So we have 4 tall and 0 short. That's what the phenotypes tell us. Right? Or you could say 100% tall, it all means the same thing. The genotypes are the letter combinations. So we're interested in what genes you have. So how many big T's do we have? So if these individuals have kids, none of them would be big T. All of them would be heterozygous. Although they're tall, right, there's no difference between these 2 phenotypes. There is a difference in what they can pass on to their children. And of course, there's no little t's. So phenotypes are what they look like. Genotypes are the letter combinations.
So what's the point of phenotypes and genotypes?
In this instance, these will give you the same. It's not about, right now, it's not about the difference on what they're going to look like, because they will look alike. It's a matter of what can they then pass on to their children. Because if you have this individual, big T, they're only now going to have children that are tall. It's not possible for this individual to have any short children. But this individual, although it's tall, they could have short children because they could pass that little tr, to be honest with you. So at this point in time, you're right, there's not a big difference. It's just a matter of what they can pass on to their children. Okay? And how, and this kind of shows how traits can actually skip generations. So you have, in the parent generation, you have a tall parent and a short parent. So you have the short gene in the parent generation. All the kids are tall. But then you could have grandkids that are short. So this sort of explains that generational gap. How you could have grandparents with the tall gene or the short gene, and then grandchildren then see the short gene. No, you always write the dominant letter first. Okay. All right, so that's one Punnett square. All right, we good with those? All right, second type of Punnett square that we dealt with. Number 2. Number 2. The second type is called incomplete dominance. Incomplete dominance. So what incomplete dominance means is that there is no particular dominant trait. This is where we get a mix, and that equals a third trait, or a third phenotype. So this is the explanation of it. I think most of you are here for this. It's like mixing paint. You have red paint and white paint. That gives you what kind of paint? Pink. Good. So let's say flowers. So we get flower colors. So big R, R's would be red flowers. Whites would be W's. So when you have one of each, you get a mix, and that's going to give you pink. Very good. So this is just a different scenario. Sometimes we have traits where you get a mix, where you don't have a dominant allele. Incomplete dominance. One isn't necessarily dominant over the other. So let's say we have a red flower and a white flower. You have a red flower and a white flower. They have kids, and again, I am pretty confident that you all can do this part. It's the phenotypes and the genotypes where we get mixed up. Phenotypes would be, 4 pink, 0 everything else, or you could say 100% pink. And the genotypes are the letter combinations. Yeah, would be 100% heterozygous. Big R, big W. So this is incomplete dominance.
So how can we do, how do you write that if you're using the numbers?
4 to 0 and 4 to 0. If you wanted to use the actual comparison, it would be 4 to 0 and 4 to 0. This is incomplete dominance. Yes. Whichever way that you would be acceptable. On the test, I will specify. On the SOL, they will specify. It just means the same. It means the same thing. Okay, but you could verbally say it anyway.
I gotta fix that sometimes. I don't want to get it.
Yeah, no. We'll specify. All right. Number 3. The third type of Punnett square is very similar to the second kind. So this is called codominance. So codominance means 2. So this is, I'm sorry, codominance means it's very similar to incomplete dominance. It just means that one is not necessarily dominant over the other. So this is where we get a mix, but we it's not like mixing paint where you would mix black and white and get gray. You would actually show both colors. Yeah.
So it's like mixing Play-Doh.
Yeah, exactly. I did. That applied to just then. So, and we said that to just make the, we were trying to, at that point, decipher what was dominant and recessive. So that was just to drive that point home. But you can have situations where you will have different letters expressed. Okay. All right, so this time, let's say we have chickens. We have black chickens. We have white chickens. And this time when we have the BW, we're not going to get a gray chicken. We don't see gray chickens. We'll see black and white chickens. All right. So let's do a Punnett square. Let's say that both chickens are black and white. Both chickens are black and white. So we do our Punnett square, which again, I'm pretty confident you all can do this part. What do we do with these individuals? You have two chickens that are both black and white. Let's look at the phenotypes.
Would it be 50% black and white and the rest like that?
Yeah. So how many black chicken would we have, Stephanie? How many black chicken would we have? One. One black. Two both, yep. And one white. Yep. And genotypes are the same way. You would have one homozygous black, two heterozygous, one homozygous white. Yeah, exactly. If you want to put that in percentages, you could say if there's a 50%, if you have a black and white chicken and a black and white chicken that have kids, there's a 50% chance that they would have chickens that are both black and white. Or a 25% chance that they're all going to be black. Or there's a 25% chance that they're all going to be white. So codominance. There's going to be a mix. Any questions with this? It's a great question. Come see me after school tomorrow and we'll go over that. Or you can watch this podcast again and it'll be good. All right, are we done here? Remember, there are 6 kinds of Punnett squares. This is only 3. I know. Number 4. So number 4 is called multiple alleles. Number 4 is called multiple alleles. And all this means multiple dominant alleles. Multiple dominant alleles. We use blood type as our example here. We use blood types. So A and B is dominant to O. And we'll symbolize O with a little I. Okay? We'll symbolize O with just a little I. So if you're type A blood, make a little chart over here, and you can write this or not write this over here if you have your packet on this. If you're type A blood, you're either homozygous A or you're heterozygous A. Either way, you're type A blood. If you're B blood, you're either homozygous B or you're heterozygous B. Again, either way, you're B blood. If you have both the A and the B gene, you're AB blood. And if you have type O blood, what two letters would you be? Only way to get the recessive trait is for both of the letters to be lowercase. That's not a problem. Half the people in the planet are O blood, so
So since O is 2R, so O is going to be the A and the B.
exactly. So if you're type O blood and you have a parent that is type A blood, the only thing that your parent can be is this. Because if your parent donates the A, you don't have O blood. You're going to have a combination of A or I don't know what the other parent is. What's that? You only have two genes that make up the trait? AB, you have one of each gene. Yep. All right. Yeah. Okay, let's do a sample Punnett square. Let's say you have an AB person. Let's say you have an AB person, and they have children with an O person.
It's 50-50.
So here are the types of questions. So you do the Punnett square. So can these two individuals have a child with O blood? No. Why not? It's not possible. Yeah. You don't see this in the Punnett square. Can these two people have a child with O blood? No. What if they do? There's not something wrong with the child. Okay. Yeah, maybe there was a hospital mix-up, which does happen. Babies have been sent home with the wrong parent, and they have to do a blood test to figure out whose baby belongs to who, or has a different father. Yeah.
A parent can be I, and I, and then O.
That's the worst. If you're in the hospital, you can tell.
So let's do one more of these Punnett squares. All right, let's say we have an A person that's heterozygous. Ladies and gentlemen, here's another situation. I'm trying to clear up some of your questions. We have an A person that is heterozygous, and we have a B person that is heterozygous. Can these two people, even though one parent is A and one parent is B, can they have a child that is type O? Yes. If the B parent donates the little I, and the A parent donates the little I, you can have an O child. It's a 25% chance, but there's still a chance. It's still possible. Last Punnett square on this sheet, number 5. Fifth type of Punnett square, sex-linked traits. Some of you were absent for this, so you need to pay close attention. Sex-linked traits. Sex-linked traits are traits linked to the X chromosome only, not the Y. Right? These are things like being colorblind, like hemophilia, like baldness. Anyone ever wonder why there's not a whole lot of bald women running around? Like they actually have the bald gene? Have you ever heard that? Have you ever heard that baldness? Have you ever heard that baldness comes from the mother's side? So if you're a male, where do you get your Y chromosome from? Yeah. So with sex-linked traits, because they're linked to the X chromosome, if you're a guy, does it matter what your father is? No. Your dad always gives you the Y chromosome. So it only matters what your mother is. So we look at the baldness gene. It only matters what men are mostly bald. We look at the population of people on this planet that are bald. It's mostly a male-dominant trait. It comes from your mother's side because that's who gives you the X chromosome. So, let's focus on this. So, linked to the X chromosome. The traits that we're dealing with are all recessive traits. So that means when we look at our X chromosomes, you're either going to be X big N and you're normal, or your X little n and you're going to have the disorder. Whatever that disorder is. We'll just call it disorder. Or we could mean colorblind, hemophilia. So let's say we have a mother who is homozygous, normal, marries a colorblind male. Can they have colorblind children? Let's take a look. A female, she is homozygous normal. So what are her homozygous normal? X big N's. Now you have a male, XY, that is colorblind. So what is his X? It's little n. The first box, X big N, X little n, normal or colorblind? Normal. This female is heterozygous and a carrier, but she's normal. So the second box. X big N, X little n. Normal or colorblind? Normal. XY, we have a male. That's X big N line. Normal or colorblind? Normal. XY male. Normal. So, can these people have a colorblind child? If we're talking about hemophilia, can these people have hemophilia? No. If we're talking about baldness, you're going to see any bald people? No. But these females could pass that trait on to their offspring. They could be carriers.
So if they meet with someone who